clap.
Yo.
Hi guys, welcome back to chapter 3 for biochemistry series, which is non-enzymatic protein function and protein analysis. In this video also we'll make a one-shot video for this chapter 3, all you need to know for MCAT. There are some things that I would go in detail which is not there on this page, so that you know everything you want to know for your MCAT. So first let's go to the structural proteins. Structural proteins are the ones that creates the structure of the cell, or the so it composes the cytoskeleton of the cell. There are different anchoring proteins and the proteins in the extracellular matrix. Okay, so it's basically responsible to make the rigidity of the structures in our body. So the most common ones is the collagen, elastin, keratin, actin, and tubulin. Out of this, I think collagen, keratin, and actin are the important ones. Collagen is just made up of 3 protein strands that are intertwined with each other, and it's basically it gives rigidity to our bones and all those kind of bones and skin and all those kind of things. Keratin is made up of 2 strands of proteins intertwined with each other, so it has 2 alpha helices, which is left-handed, and that, if I'm right, it ends up making a right-handed protein structure. Actin, if you see in your biology review, actin is the thin filament of our muscles in the sliding filament theory. So yeah, I think these 3 are the most important in the structural proteins. Motor proteins, motor proteins are the one which helps things to move in our body. So it has 1 or more heads, which is capable of making a force or some conformational change. So, and they're generally active, so it needs catalytic activity like ATP pases, that is just cleavage of ADP so that they can use the power from the mitochondria to do a power movement or a conformational change. Okay, like muscle contraction in your body, vesicle movements in the cell motility, all those kind of things are done by motor proteins. It's kind of a steering wheel for the cell. So these are myosin, which is the thick filament of the sliding filament theory, kinesin, dynein. These are found in flagella of the bacteria or different cells, cilia, all those kind of things, which helps in the motility of some things. Okay, binding proteins. So binding proteins are used to bind things, as the name suggests. So it binds to a specific substrate, either to sequester it in a body or hold its concentration at steady rate. Okay, so usually you see it in immunobiology or those kind of things, that you will have antigen binding proteins or those kind of things, antigen presenting proteins, which will bind to the protein and it will either sequester it, phagocytosis, those kind of things. Binding proteins help it with. Phagocytosis, if you don't know, it's just if there's a microbe in your body, the binding proteins will detect it, and if it's not and if it is detected not as a part of our body, it will just phagocytosis and then just make pores in it, pucker it, and it will just burst out and get killed. Okay, cell addition molecules, or CAM. This is very high yield topic. You should know what are your cell addition molecules. So one is cathedrins, which is calcium dependent glycoproteins, which helps in putting the cells together. So adhesion means putting 2 things together, like sticking 2 things together. So cathedrins do it because of the calcium. Integrins are double membraned, so it's like two membranes. Oops. Yeah. So why is not working? Sorry about that. So it's just like double membranous structure. Okay, it's not working for now. Anyway, so it's just a double membranous structures which permit cells to adhere to the proteins in the extracellular matrix. Okay, selectins are also similar kind of helps the cell to adhere to each other with the help of the carbohydrates like glucose and other things which is present. So for example, if there's a cell which has like glucose on it, so another cell with selectin will come and bind to that glucose and hence binding the two cells together. Okay, this is also very common in immune system. Antibodies are immunoglobulins, which has like four chains, two heavy and two light. It has antigen binding site, which binds to the antigen. That is called the variable site, and it has one of the constant sites. I can show you the picture of it. Immunoglobulins. So if you go to images. Yeah, so as you can see here, these two are the light chains, these are the heavy chains, these are the variable regions where antigens can bind, and these are the constant regions. See, in this one it's more clear. So variable regions, constant regions, and these are the heavy chains. The full chains are the heavy chains, and these small chains are the light chains. Okay. Coming to the biosignaling, so proteins are used to signal different other cells for specific things. Oops, battery sign down. So it can be used for regulating ion flow into or out of the cell. So it could be like on the membrane, like this. So this is the membrane. So it could be on the membrane and can allow in and out movement. Some are ungated channels which are like always open. Mostly there's like potassium channels, calcium, somewhere there's like calcium channels which are always open. Voltage gated channels are usually what do you say? Oh, shit. I'm losing words. Okay, so voltage gated channels are usually restricted by the membrane potential. So if there's like a specific potential, then only it will open. Otherwise, if it's a higher potential or lower potential, it won't open. Ligand gated channel is like if there's a specific ligand which is coming and binding to it. For example, if you see epinephrine is coming and binding to the G protein coupled receptor. So these this is like the ligand gated channels, so open in the presence of a specific binding substance. Enzyme linked receptor is participate in cell signaling through extracellular ligand binding and initiation of secondary messenger cascades. So for example, now example of enzyme linked receptor is a G protein coupled receptor. So first something will come and bind to one of the enzyme, and that enzyme will activate another enzyme which will create a secondary messenger, and that messenger will go to the target area or the cell and do the changes. So as you see here, the G protein coupled receptor mechanism. So here the epinephrine is coming, binding to the G protein, this G protein coupled receptor, then this G protein coupled receptor turns the GTP from GDP to GTP, and this GTP will bind to the G protein. This G protein will become active, and then another enzyme, adenylyl cyclase, will convert ATP into CAMP, which is cyclic adenomonophosphate. So from adenotriphosphate, it will become to cyclic adenomonophosphate, which is also a second type of a secondary messenger. And that secondary messenger will then go to a target protein, which is kinase A. And as we learned in the chapter 2, kinase A will result in adding a phosphate group. So that addition of phosphate group can inhibit the glycogen synthesis and and promotes the glycogen breakdown, because epinephrine is also a fight and flight hormone, right? So in that thing, we won't store sugar, we will use your sugar. So yeah, and this process is catabolic or anabolic. Yes, it's catabolic because we are breaking our glycogen to get energy and use energy. So in catabolic, this will make it promote, and then this will make it inhibit. Okay, that's what we learned. Now, how can we isolate protein? So we know there's a primary structure where there's like a big amino acid chain. So how can we know that which protein is which? So by knowing the primary structure, we will know what protein is what, and to know that, we have different techniques. So one of the techniques is called electrophoresis. Electrophoresis use a polyacrylamide gel, polyacrylamide gel to separate the amino acids from each other. It's like a gel matrix, a mesh kind of a thing, which will separate all the proteins and amino acid from each other. So a type of electrophoresis is the native page. So page means polyacrylamide gel electrophoresis. That's the full form of page. So native page is this thing that maintains the protein shape. It does not break the protein or doesn't change the mass or the charge of the protein. The protein is intact. That's why we can just take that protein again into use for some other experiments, because we are not denaturing the protein, or we're not doing anything with the protein. But sometimes it becomes difficult to analyze because all the proteins will have different mass, different charge, so it's difficult to compare with each other. It's like you have to compare watermelons with oranges. So one is very big, one is very small. So a better way of doing it is SDS page, which actually denatures the proteins and make it a specific charge and size so that it's more accurate. Then we are just comparing oranges with oranges, not watermelon with oranges, right? But because we're denaturing it, the functional protein will be, destroyed, and we can't recapture it. SDS is the full form is SDS pages. Sodium didosyl sulfhydryl page, which is polyacrylamide gel electrophoresis. So what this SDS does is SDS breaks the protein and puts a negative charge on it so that all the charges are same and the size is same. And we can do the electrophoresis on our own. I can show you the picture of it. So this is how it will look. So this is the glass slab or the mesh-like structures. These are the glass wells, and this is the power source. And always remember, it goes from cathode to anode. So what's a new point in it is if you see here, there's the negative pole, and here you have the positive. On your left hand side you have the positive pole. So generally we know that cathode is positive and anode is negative, but here the negative is the cathode and positive is the anode. Okay, so that's very important to know. Then we came to isoelectric focusing. It is very similar to electrophoresis. It's just the difference is it separates according to the isoelectric point, like we did in chapter 1. So similarly, you will have the mesh, and you will let it go from negative to positive, and wherever and it stops wherever it gets its pH is equal to pi. So this is the point which it's looking for. Okay, pH is equal to pi point. Then we go to chromatography. Chromatography you would have done in your organic chemistry labs and all those things. So chromatography, you will have like a chromatograph paper which has like polar coating on it, and if you put like a dot of your protein and you let it like go in an alcohol or any other solvent, wherever it's like stopping, you will know that how polar that thing is and how non-polar. The more it goes up, it will be non-polar, and the less it goes, it will be polar. Okay, because stationary phase will be polar and the mobile phase, which is the solvent, will be non-polar. So polar will bond with the polar one, so the the more polar protein will go smaller distance because it will just bind to the stationary phase, whereas non-polar will bind with non-polar and it will go up with the solvent. It will just go up. Okay, now we go to the column chromatography. Column chromatography, it has like small beads of polar compound as the stationary phase. So if you put something, whatever is polar will bind with that beads, and whatever is non-polar will come down. Ion exchange chromatography is like of two types, cation exchange and anion exchange. If it's anion exchange, your bead will be positive charge and all the anion, which is negatively charged, will bind to it, and you will have all the cation or the positively charged protein out first, and all the negative charge protein will bind in the anion exchange because anions are getting exchanged. In the cation exchange, it will be your beads will be negatively charged, and when you put the protein, or the positively charged protein will bind to the negatively charged, and what first comes out is your negative charge. Okay. Size exclusion chromatography relies on the porous beads. Sorry. Size exclusion chromatography relies on the porous beads. It's many people think that the larger ones will get stuck and the smaller ones will come out, but that's not true. Beads will have like small holes in between it, so the smaller proteins that fit in that size will stay and the larger ones will come first. Okay, so larger molecules will elute first and the smaller ones will get stuck in the beads. Affinity chromatography is like a ligand kind of thing. So you will put beads which will have high affinity to some ligand which will be present on the protein. So first you will mark the protein with the ligand, and then you will put that ligand will bind with the beads, and those protein will get stuck in it, and the other proteins which does not have that ligand will not bind to that protein and elute first. That's what's written here. Using a bound receptor or a ligand as an and an elute with free ligand or a receptor for a protein of interest. Okay, they don't do a very good job explaining it, but I think you understand it, that the beads will have high affinity to one ligand, and we will mark all our proteins that we need to get bonded with that beads. We will specifically put the ligands on that bead, and then we will put that in it. So whichever protein will have that ligand will bind to that beads, and whichever does not will elute first. Okay, then coming to protein analysis, how we will know like what amino acids are there in that protein. So the most used technique is X-ray crystallography, that we will make like small crystals of the protein, and then we will put X-ray light through it, and wherever it puts the light, we'll put like multiple observations, and then we'll give everything to the computer, and computer will tell you that, okay, this is the structure of the protein. So this is how X-ray crystallography works. Another thing is NMR. NMR you would have done in your labs. So NMR also helps in determining the structure using the hydrogens and the carbon atoms. Then amino acid sequence, how to know a specific sequence. So in X-ray crystallography and NMR, we just denature the protein. So we know what amino acids it have, but we can't know the entire sequence. So to know it sequence-wise, we do Edmond degradation. One bad thing about Edmond degradation is it can't have like large proteins. So if you want to do a polypeptide kind of thing, you have to break it into small So we have to break the polypeptide into oligopeptides so that we can run it through Edmond degradation. So here it will bind to the N-terminus of the protein and break one amino acid, and then we can see, okay, this amino acid is this amino acid is this, and we can know the sequence. Okay. Then we come to the concentration. We can know concentration either by calorimetry, UV spectroscopy, or the color change. The most used one is the Bradford protein assay, which is important for your MCAT. And the dye it uses is called... sorry, I don't remember the name. Yeah, Kumasi Brilliant Blue Dye. So make sure you remember it. So Bradford protein assay use Kumasi Brilliant Blue Dye. So because it's blue, you can remember the color change. It's from brown green to blue. Okay, so it goes from brownish green color to the blue color. That's why it's called Kumasi Brilliant Blue Dye. Okay. Then if you want to check the absorbance of any protein, we use Beer-Lambert law, which is absorbance is equal to epsilon times concentration times the path length. Okay. What else? Do we have something else which is not covered? I think pretty much everything is covered in the chapter from this page. The only thing is this part. I did try to explain you pretty good, but if you still have some questions, just put in the comment section. I'll be happy to answer. I'll try to make separate videos on electrophoresis, isoelectric focusing, and chromatographies later, but for now, I think you can just learn what I told you, and I think you'll be good for your exam. All the best for your exam. Do good. Have some rest, and don't do all-nighters. It does not help for me, and I think it won't help for you also. Focus on the content review, and then try to do as many question papers as you can. And yeah, all the best. Have a good day. Bye.